What are gibberellins? Gibberellins are plant hormones that regulate growth and influence various development processes, including stem elongation, germination, dormancy, flowering, sex expression, enzyme induction, and leaf and fruit synthesis. They are present in especially high concentration in young leaves and seeds, as well as stems, where they have an important role in determining their growth. How do gibberellins promote stem elongation? The height of some plants is partly controlled by their genes. However, applying active gibberellins to plants which would normally remain short, such as cabbages, can stimulate them to grow. As yet, little is known about how gibberellins cause this effect. It is thought that gibberellins directly affect the division, elongation, and differentiation of the plant cells. Giberellins and seed germination. When the seed is shed from the parent plant, it is dormant. This means that it contains very little water and is metabolically inactive, thus allowing the seed to survive in adverse conditions such as winter and only germinating in favorable conditions such as spring. The seed's embryo, which will grow to form a new plant when the seed germinates, is surrounded by endosperm tissue. Endosperm is a food store containing starch. On the outer edge of the endosperm is a protein-rich aleuron layer. The whole seed is then covered by a tough, waterproof, protective layer. When the seed absorbs water, this stimulates the production of gibberellins by the embryo. And the gibberellins then stimulates the synthesis of amylase by the cells in the aleuron layer. The amylase hydrolyzes the starch molecules in the endosperm, converting them to maltose molecules. These are then converted to glucose and transported to the embryo, providing a source of carbohydrate that can respire to provide energy as the embryo begins to grow. Gibberellins cause this effect by regulating the gene that synthesizes amylase. What is abscisic acid? Abscisic acid is a single compound unlike the auxins, gibberellins and cytokinins. It was called abscissin 2 originally because it was thought to play a major role in abscission of fruits. At about the same time, another group was calling it dormant because they thought it had a major role in bud dormancy. The name abscisic acid ABA was coined by a compromise between the two groups. Though ABA generally is thought to play mostly inhibitory roles, it has many promoting functions as well, such as bud dormancy. The newly developing leaves growing above the meristem become converted into stiff bud scales that wrap the meristem closely and will protect it from mechanical damage and drying out during the winter. Seedling growth. ABA inhibits the growth of seedlings. Apical dominance. ABA moving up from the roots to the stem synergizes with auxins, moving down from the apical meristem to the stem, suppressing the development of lateral buds. The result is inhibition of branching or apical dominance. And what we are going to focus on closely closing of the stomata and abscission. Closing of stomata. If a plant is subjected to harsh environmental conditions such as high temperatures or reduced water supply, it responds by secreting ABA. In a plant in drought conditions, the concentration of ABA can rise to 40 times that which would normally be present. This high concentration of ABA causes the stomata to close, which reduces the volume of water vapor lost due to transpiration. Each guard cell has a relatively thick cell wall on the side next to the stoma and relatively thin wall on the opposite side. When the guard cell becomes turgid, they expand into a curved shape because the thick cell wall resists the expansion more than the outer wall. This leaves a space between them, thus the open stomata. When the guard cell loses water, they become flaccid and collapse together so that the stoma closes as shown in the diagram. Abscission. Trees regularly drop their leaves at certain times in a year. For example, autumn. The leaves fall because the leaf stalk or petiole breaks off from the stem. First, useful substances are withdrawn from the leaves and taken back into the stem. This involves the breakdown of the pigments in the leaves, which results in the changing of their green color to yellow, golds, and reds. An abscission zone forms where the petiole meets the stem, which consists of two layers, the separation layer and the protective layer. The separation layer is made up of small cells with thin cell walls, and the protective layer is made up of cells whose walls contain suberin. Enzymes then break down the cell walls in the separation layer, and the petiole breaks at this point. The protective layer remains, forming a scar which can sometimes be seen on the stem where the leaf used to be.
There's a set going strong, a lot of things going on. The man of the hour has an air of great power. The dudes have envied him for so long.